right. I'm Dwight Seal. I don't know if anybody knows me or not, but I used to come speak to the beekeeping group every once in a while. But my former life, I was worked for the Department of Agriculture in the pesticide regulatory program. Give you a brief history. We used to give exams. We had a guy come in one day and he wanted to take a research exam that lived in Dobson. I was like, we don't have, why we got a guy wanting to take a research? That's for smart people. So it was Gary, Kathy's husband, that was taking the exam. So that's how he and I met. So it sort of formed into a pretty good relationship. And then his wife, Kathy, we're getting ready to introduce her. She, Dr. Katy Lawrence. Both of these people are researchers. Gary was at Mississippi State University, retired 6 years ago, 7. All right, and Kathy, Dr. Katy Lawrence, is still currently employed at Auburn University, working out her last days of sick and annual leave to officially retire. Both of these are researchers. They're nematologists, and she'll get into more about what nematodes are in a moment. But anyway, Kathy is she's with the Department of Entomology and Plant Pathology. She's a professor of nematology, and that's, like I said, in Auburn. She got her undergraduate at New Mexico State, her master's at Mississippi State, not Mississippi, right?
Right.
Okay. That's fighting words.
Yep.
NC State and Carolina are sort of similar. But anyway, Kathy and Gary both currently have their own private research company in, I guess you call it the Beulah. You consider that Beulah there?
I think it's Dobson.
Dobson, Beulah area, where they do research on other crops. But anyway, she's telling me about she had a graduate student that did research on entomopathogenic nematodes for the small hive beetle control. And I think she oversaw the research, and she's going to teach us about that tonight. So we want to kill the small hive beetles. All right?
Thank you.
Thank you for the invitation to talk to you all tonight. I am a plant pathologist, plant diseases, and we specialized in nematodes, which are one of those organisms that cause disease. So if you guys all have a garden at home, can you guys hear okay? Okay, good. On your tomatoes, do you ever pull up tomatoes and you see galls on the nematodes, galls on the roots? Those are plant parasitic nematodes, and Gary and I work with those all the time too. That's most of what we work with is the plant parasitic nematodes. But I did have a graduate student back in 2000 who wanted to work with entomopathogenic nematodes. So we got with our bee scientist and small hive beetles, and I'll show you some of the research that she did today.
But I did give you my email if you want to call, and I sent the paper to Dwight, and I think you've sent it out to anybody who've wanted. Entomopathogenic nematodes. So insects are called entomology. So that's the ento part of the word. Pathogenic, that's our pathogen. So the nematode is a pathogen of insects. It's a nice big word, but that's what it means. It's a pathogen of insects. And you can see them on the left-hand side. Now these are tiny. So look at your strand of hair. That's how thick they are. So they're very tiny. So we're talking the microns, very small. But the insects they attack are all the soil-dwelling ones. So any insect that's in the soil are the ones that they'll attack. And you see the one on the left, a regular June beetle grub, and the one on the right is brown. So white is healthy. The brown one has been attacked by nematodes and it's dead. So it's very visual to see when that occurs. And so these nematodes I consider beneficial. We work with a lot of pathogens, but these are the beneficial ones that attack insects. So our beneficial nematodes are always attacking insects and bringing our nematode numbers down. So this would be an organic production too, if you're going that direction. Just so you see the infective stage, so it's worm-like. It's almost 1 millimeter long. It's only less than 0.3 millimeters wide. But the identification on these, they're soft bodies. You think of earthworms, these are smaller. And the difference is these are non-segmented. So earthworms have segments. These do not. They're basically a tube inside a tube is the way we define it for class. Gary smiles. They eat and reproduce. That's all they do. So it's a eat and reproduction tube inside a tube, and that's what keeps them going. Their life cycle is very short. So you sometimes hear them referred to as roundworms. They're obligate parasites of insects. This is important in that they are obligated to feed on a living insect. So I can't grow them unless I grow them on insects. So I really like fungi that I can keep growing on pea media and potatoes. Don't step, okay. But these obligate parasites on insects, I have to feed them an insect to keep them growing, which is why they're kind of expensive when you buy them. So that's the idea there. So they're great. They have to live on an insect to complete their life cycle, so they're constantly after living insects. Great biological controls. They are out there in the soil all the time. And in fact, I extracted some soil today, and Gary was working on a microscope we could bring, but our light bulbs were out, so we didn't bring them. But they're always naturally. So they're out there all the time. We're just trying to increase their numbers and put them in the right place so that they can benefit. That's what we're trying to do. And you see, they're in the soil. They always live in the soil. So they don't have eyes, but they locate by breathing the carbon dioxide. They can find the insect by detecting carbon dioxide, but also any chemical cues. So, or vibrations in the soil. They respond to those. So that's how they find the insects. There we go. And you see this insect larvae? And then you see all the little white squigglies? Those are the nematodes that came out of this larvae after it died. So they can produce millions of them very quickly. So in less than 2 weeks, these nematodes pass through several generations inside this nematode cadaver and consume that insect. So it's like these insects, yeah, they do. They enter the insect, they consume the inside and kill it, go through lots of generations, consume all that food source, and then they escape to go look for the next one. So it's a great biological control, as you can see there. So you see the little white ones, the nematodes, and you can see that size versus what a larva grub would be. So it's a nice line drawing of the insect. This is off one of the sales pages. So you can see, if I can make that mouse work. No, and I can't go that far, can I? The infective stage, the IJ is your infective stage. They're out in the soil. They sense a juvenile or a larvae, a grub somewhere. Any opening is fine. So they can go through the mouth, the anus, any pores in that insect. Once they get in, these guys, they also carry a bacteria with them, which they release. But that infected larvae will turn white to beige to brown, and some of them are red within 2 to 4 days. Oh, I have, thank you. Within 2 to 4 days after infection, and then they become kind of slimy, but because the nematode's carrying a bacteria. And then the dead larvae will disintegrate and die, and the nematodes reproduce several generations till there's nothing left. They consume it completely, and then they escape and go find the next one. So that's how their life cycle works. Very nice, very neat, and very quickly. There are 2 genera, so if you buy these, and you can commercially buy these. There are Steinernemas, and there are Heterorhabdites. Both of them are out there. There's lots of species within each one. So we had better luck with Steinernema in our research in Alabama, but things can vary. So there are Steinernema and Heterorhabdites. Steinernemas are males and females, so it takes a larger population when you have males and females to mate, lay eggs, and there are more numbers. You're going to have larger numbers. Heterorhabdites are hermaphrodites. They are male and female within the same body, so it takes fewer of them because they're going to reproduce. The whole cycle of reproduction is what these nematodes are trying to do. So that's what we begin with. And those are the 2 genera. Nice scanning electron microscopy. I love the pictures. This is the head of a Steinernema that you can see there. It's an extremely capable killer. This is with a graduate student. This is one of her slides she put together. 2 of these Steinernema, the male and a female, are enough to enter an insect and incubate and turn him into spawned worms. It only takes 2. Parasite partners with Xenorhabditis is the bacterium. Steinernemas and Xenorhabditis always work together, but as the evolution has occurred over time, the Steinernema species and the Xenorhabditis species are always different. So there's not a specific one always. There's always a specific species that matches in the pairs. They've evolved together. So the bacterium is passed by the nematode, and the nematode puts it into the insect, and the bacteria starts to generate and dissolve the insect contents, and then the nematode and the bacteria both proliferate. So this is a symbiotic relationship between the nematode and the bacterium to kill the insect. So you have two of these organisms that take them out. It's amazing how these develop. Life cycle. Can I move okay? Okay. So we got our infective juvenile at the top, finds that larvae in the soil, penetrates it, releases the bacterium you can see in the hemocoel, and then between the bacteria and the nematode, they kill that host within 2 to 4 days. So your insect is dead within 2 to 4 days. The bacteria start to proliferate. Bacteria grow, reproduce in 20 minutes. So it doesn't take them 24 hours to go from 1 to 1,000,000. The nematodes take a little bit longer. They take a few weeks to get through a generation. So the bacteria grows, then the nematode. And this is the life cycle of a nematode, which is very much like an insect. It's an animal, so it lays eggs. It goes through 4 juvenile stages like an aphid. So you know the aphids shed their skin and molt 4 times before they become an adult. Nematodes do the same thing. And if you look between 2 and 3, it's called an infective juvenile. And that one leaves the insect's body and goes hunting another one. So between the 2nd and 3rd stage, it becomes infective. It leaves that insect and it goes in hunting the next one to kill. And it always is already carrying the bacteria with it. So they go in that cycle. Some of them stay in. And this is where you see the Steinernema. We have males and females. That's my easiest way to try to identify them. The Heterorhabditis, they're both within one body. So that's our best chance. When Gary and I are trying to identify, do we have males and females or just one? So you can keep the species, or the genera different. So that's the general life cycle. Nematodes take a couple of weeks. The bacteria is very quickly. It goes very fast to take out your insects. This is your beetle, or this is the one that Wendy worked with. Small hive beetle, Aethina timidae, that's the adult. So that's the one we worked with in Alabama. I'm not the entomologist, I'm the nematologist, so I don't know if yours was the exact same species. It is, okay. On the top, you see the backside, so you see what the beetle, the pupa, and the larvae look like. The bottom is the reverse, so you see the bottom of them all the way across. So that was the insect we were after. So for the entomopathic nematodes, we've worked with lots of these. They kill over 200 species of insects. So there's 100 different families. So Gary's worked with the entomopathic nematodes on soybeans, on loopers. We've worked with them on turf, on grubs. So working with them on the small hive beetles. These are options that you can always do. These are interesting characteristics for these nematodes because they're in the soil and they're hunting insects. Steinernema and Heterorhabditis do it differently. So Steinernema does ambush. So they sit and wait until the insect comes by and then they ambush him. So that's their hunting style. Heterorhabditis, on the other hand, is a mover. So he's considered a cruiser. That one moves through the soil hunting the insects. So they work in different ways. So often we put these together, trying to get all your insects. So that may be one reason why you want to do a Steinernema and a Heterorhabditis, because some ambush and some cruise. You just want to kill all the insects. I just thought it was neat they did that. Okay, this is from Wendy's. So she worked with the honeybees and the small hive beetles and our entomopathic nematodes, and I was going to show you some of her data. She talked about what causes colony loss, and I know there's lots of different things. So we focused on the very bottom thing, the hive beetles, and that's what she was looking at. Interesting, this is from 2020, so I don't know if it's changed. But the small hive beetles are considered a secondary pest, native to Africa. So they actually came from Africa, reported in the US in Florida in 1998, I believe, 1998. Also a report from Charleston, South Carolina from 1996 and 97. Difficult to manage. The distribution is what there's some differences in the literature there. Some say it's across the entire world except for Antarctica, and others say it's only in these red spots. So it's probably everywhere, but the literature has both of these. But it started in the US in 1998, so it's got a good foothold. And you notice it's across the whole country.
If it helps, some Canadian beekeepers do not deal with
Okay.
the small hive beetle.
So they do not occur up there. Okay. And we're just checking literature to see what's there. Okay. Life cycle of the small hive beetle. And this is from Wendy's work. You start with your hive. You have the beetles inside the honeycomb, the hives inside. They mate, relay eggs, and unfortunately, we looked these up. These beetles eat everything inside the beehive. So the eggs will hatch into the larvae, which is the cycle that goes down into the soil. And once the larvae is in the soil, then the entomopathogenic nematodes will attack the larvae and the pupae and will kill that stage. But if we can kill that stage, we don't have adults. So that's what we're after. So we can kill the larvae stage. But you can see in this picture on the side where you have the beetles inside the honeybees. So you see how tiny, they're pretty small. But that's the general life cycle. So we're hitting the larval stage. This was the current management in Alabama. So with all these cultural, mechanical, and chemicals, and I don't know if those chemicals are still the ones because it's been 5 years, but we worked with the biologicals, and that shows you the Heterorhabditis and the Steinernema and the product we bought. So the one that we bought one online to test, and we bought several of them to test, and that's where we got our entomopathogenic nematodes. And with that, I was showing you, this is a nice life cycle that came from the Arabica where I bought them. So they put this in there. So where you have your small hive larvae enter the soil. You can see the cruiser nematodes and the ambush ones. So the cruiser is Heterorhabditis, just cruising around. The Steinernemas tend to stay in a group. They enter that larvae host, and then it doesn't take 2 to 4 days and it dies. Then others are attracted to that host, and it becomes a feeding frenzy for the bacteria and the nematodes. They go through several generations until it bursts through the insect and all the contents are gone, and then they go back out looking for more to eat. So that's the basics on that one from the company that sold it. So here's our Steinernema. That one, there's 95 known species of this nematode. So there's 95 different ones. So there's a lot out there. We tested 4, but there's 95. That was what you could buy. So the Steinernema feltii was one we could purchase, and we tested that one. Steinernema Rio Bravo was another one we could get from the Arizona company, and we tested it. Cruzii was one, and then Carpocapsii were the ones that we could buy. So we tested those species because that's what I could purchase. Then we also, with these guys, it's always a Xenorhabditis bacterium that comes with them. But the bacteria species changes with every one of those species of the nematodes. So they all change slightly. Heterorhabditis, that's the ones that are hermaphrodites. But in following generations, we notice there can end up being males and females. So that one is slightly different. 16 species, I could buy 2 online. So Bacteriophoria, I could purchase, and indica. So those are 2 species that I could purchase. Now you can't always buy these everywhere. I know my Hawaii colleagues have to grow their own because they're not allowed to bring them in. So you end up with, there's lots of different species in different areas that you can look for. And the bacteria for Heterorhabditis is called a Photorhabditis species that goes with it. So they're very specific, but they come with them in the nematodes. There are differences in color if you're out looking. So the Steinernema, the carcass is the top one, is more of a brown color. The Heterorhabditis makes more of a red color on the bottom. So that's a visual that you may see differences between those nematodes that are in there. And this is some characteristics if it makes a difference when you're looking at these nematodes. So you notice as you come through the bacterial symbiotes, and this is where you see the Steiner Nematodes has a Xenorhabditis nemophagus. That's its species. Rio Bravo comes with a different cannibalism. It was a nice name for that one. So all the genus species match and are specific for the nematodes. But you also see the hunting techniques. And then the other part on these is you're putting them out. Desiccation. Nematodes live in the water moisture around in the soil, so they can't dry out. So we're always trying to keep them moist. So today would have been a good day. Most of them, some can tolerate some drying out where others can't. So you see Capricacii, Carpocapsin can hide desiccation, so it can dry out more than Cruzii cannot dry out. So if you want to take in those considerations when you're picking them out. Ultraviolet light. Excuse me, the day the sunlight gets into the problems, they can't dry out, the light will break them down. So heat and cold, and their natural habitat. So it's interesting that Feltii comes from shaded grounds. I like that Cruzii came from conifer forests. The Carpocapsi, which did best in our test, was more of a sandy loam, which was the soil we typically had. So you see, but the Heterorhabditis does fine in a heavy clay. So if your hive bees are on a heavy clay, that may be a better option. So those are just characteristics of each type. And then when I looked here too, the moisture and the temperatures. So Carpocapsi did well for us, 40 to 90 degrees. That's pretty much our area. We're usually pretty warm. Others were cooler temperature areas. So Feltii was more about 60 degrees. Might do better in Canada. So different ones in different areas. And different soil types. So they have done research on which ones they do better underneath the hives. So in Alabama, we had 3 different soil types that we really worked with. So if you look at those, the KLS is a sandy loam. It's a loamy sand. It's one of our general everyone's. Up north, the DLS was the Decatur silt loam. It's a very heavy clay soil. So that was where we have some. And down in the south toward the ocean, it's a very fine sandy loam. So we were testing all these different soil types to see. So she had a lot of objectives, and this is the way the experiments worked. You see the top picture? Those are the small hive larvae. So that's the larvae we would increase and get them. And then underneath it is a petri dish. So a petri dish is just a little round plate. It's only about that big. Filled it with the soil. And then you see the 5 larvae she put in there? So we was trying to see how we could improvise what happens in the field. So that was what we were up to. So she did the petri dishes. She added 5, 10, 20, and 40 larvae to each petri dish. We did 10 replications, put them in the growth chamber. 25C is their happy point, it's about 75 degrees. 80% relative humidity, gave them 20 days, did they pupate? So we're just making sure that the life cycle would go through with our experimental conditions. And it worked. So you can see 5, 10, 20, and 40 larvae. They did pupate with their average numbers about 90 degrees, about 90% pupated, until we got to 40, which got to be too many in our petri dish. So that was too big of a population. You'll notice too, blue is autoclaved. That's just pressurizing. So the same thing is in the soil. If I pressurize it, like canning green beans, I take everything out and my biologicals work beautifully. There's no competition. You can't do that in everyday life, but I can tell if something might work if I take everything else out. So that's why we do that. The natural is the orange, and we just left the soil naturally. So see here, these the larvae pupated just fine with all the natural soil competition going on. So that's where she was just checking. Then she went on and decided to do the rest of the test with 5 of the larvae. And we went in. And this time she looked at Feltii, Rio Bravo, Cruzii, Carpocapsi, and then also the Heterorhabditis, Bacteriophora, and Indica. So we looked at different species that we could buy. And that's, at the time, what we could buy. So we bought all those. She did them with 5 in the dishes, added the nematodes at various inoculum, depending how many were there, moving it up across. And different population densities. She looked at the densities. We did see at this point, if we have 40 of the larvae or 80, we tend to get more of the larvae were parasitized. So the nematode was able to find it and kill it. So the distance, the nematode is 1 micron. They don't move very far. So it's got to be pretty close to the soil. So they will go across my petri dish in a day, but they're not going to go 30 feet. They'll do 30 inches, but they won't go too far. So the more insect larvae we had, the more nematodes were able to find them, colonizing, and kill them. So that 5 was fine for growing them, but 40 was better for more kill. So the higher population of the insect, the more kill you're going to get. Make sense? And then the different species, and you can see the difference there. So for our small hive beetles, Carpocapsae killed 60 to 70%. All the other species killed 10 to 12. So there's definitely a species specific in our situation. So in these, made a lot of difference. So that was the one that we focused on is doing most of the kill in our nematodes. So you'd want to check which ones. And this is the line drawing of more of the same data. So the blue is autoclaved, the orange is natural. This is the population with 5, 10, 20, 40, and 80. But you notice by the time you get to 40 and 80, we have almost 90% kill in autoclaved or natural with Carpocapsae. The other ones here don't go up that high. So our kill numbers, if you look at that scale on the side, 14% is the highest for indica. That's a whole lot less. For Rio Bravo, we had up to 30% kill, but that was it. So Carpocapsae was a much better species for taking out our small hive beetles. And it did a pretty good job. Even if there were very few, it got 70%, but we had higher populations, it got up over 90%. So that one was the best killer that we could get. And on this one, loamy soil, she was looking at that. So this is bar chart. The bar charts are the same line charts, so you can see that. So visually, you can see Carpocapsae took them out quite rapidly and very well. And those are some nice pictures of them. So in the center, can you see the larvae and the Carpocapsae and all the nematodes coming out? Yeah. You didn't, we saw some on indica, but here we didn't find as much on Rio Bravo. But nice pictures of them. Populations. So she's got her various populations, so you can see the statistics going with it, going up from 10, 40 to 80. So the more insects, the more kill we had. So that just worked out for every species. More insects are there, the more they can find them, the better they can kill them. Did I go the wrong? Then she went on to the different soil types. So same with these 3 different insects, the entomophagous nematodes, and 3 different soil types. So the first one, the B, FSL, fine sandy loam, very much a sand. That for us, that's down at the beach, very much sand. The DSL is the silt loam, very heavy clay soil, it's 50% clay. Grows some great crops, but it's very heavy. And then the loamy sand is the last one, which is in the middle of the state. So you can look, the soil type did make a difference. So the nematodes need to be able to move through those soil particles to find the larvae, and it may affect the larvae's movement as well. So the loamy sand, I think I hit the button and it comes up, had the best kill, the highest numbers. But that soil is more of an optimal crop production soil. And the very fine sandy loam, a large soil particle so the nematode and the insect can both move. But the silt loam, which can produce some 4-bale cotton for us, did not allow the entomophagous nematode to move very well. So it didn't do as well. So whatever your soil type under your beehive may affect whether or not this works well. So if you know your type of soils, do you have more silt or more sand, will make a difference. This is a higher population, so I didn't point that out. If you do a higher population of the nematodes, you get more kill, same thing. So she went with a high population of the entomophagous nematodes, and the other one was the low. So with a high population, you can tell our kill rate was very high. We're getting close to 90% above. When we went back, I'll go back, I'm sorry, this one was only 10%. So if you didn't have, you stretched your nematodes, made them go farther, 10% is a little too low. We didn't get as much kill. But with 80%, you have almost 90% kill of everything. So that was good. But you know, we're always trying to see how far we can take it back so it doesn't cost quite so much. Okay, so from her research, the Steiner Nemo capicarpsy was the best one. Higher populations killed better, and we did get very much promising control for the Alabama. But everything we did was in the growth chamber. It was not outside. So she graduated and moved on before we got to go outside. But that was the point at the end. We did believe that species, Carpocapsae, was much more effective than anything else we bought. So if you buy these guys, 10 million nematodes will treat, I got this offline, 3,200 square feet. So it takes a lot. 50 million is half an acre, so however big you, yeah. 100 million treats an acre, so it's a lot. We bought ours from Arabica Organics online. They're out of Arizona. So that's just where I was able to find them and get them in a short amount of time. It does take about a week or two to get them in. There is also, I looked yesterday, these change frequently, but online, Grow Organic is a company, and that's their webpage. Bugs for Growers wasn't there a few years ago, but it is now. So there's several different web pages you can look at, and that's why I was just looking to see. But I bought Arabica Organics just because I think that was one of the few ones that we could buy all those different species from. But they're expensive, so that's why I was gonna, am I in the way? Let me let you look. That's why I was typing it down. 5 million was $67, and then there's shipping. These things can't get hot, so shipping's gonna cost. Gotta come in an ice chest. 10 million was that lowest one, so see that was 88. 50 million, it takes it up to where if you need 500 million, that's $735. So they're expensive. And they're organic biologicals. So these things are alive when they send them to you. So you have to be careful with them when you're applying them. But that's the cost, just so you see them. When I put them out, we put them out. So they're in a powdered medium that dissolves in water, kind of. It likes to clump up. So I like to put it out. All our biological work Gary and I do, we put them out with watering cans and water them in just to make sure we can spread them like we need to. The sprayer that we use all the time in the gardens, you have to take every screen out and use the largest nozzle you've got, or it will clog up. So that's why we often use the watering can. But you can spray them. Just take all the screens out, keep agitation going. So if you've got a backpack one, just keep wiggling. You got to keep it in solution because it'll fall out. So you can see the powder they come in. That, I think, was just the smallest one. So that was $67. Yeah. You don't have to use a lot, but yeah, they're expensive. So we water them in with lots of water. So you probably haven't seen us out in the field, but we'll take whatever biological we're testing and run down our row and water them in. So we can usually get 20, 30 feet, Gary, in a gallon of water and keep putting them out. So we do that a lot. So the sprayer, make sure it's going to clog up. Keep it agitated, take every screen out, use the largest nozzle you've got to make it go out. Continuous mixing. So that means you're wiggling with your backpack, keeping it mixed. Soil's got to stay moist. And this is with any biological I use anytime, whether it's a nematode or a bacterium or any of them. Got to keep them wet. They need moisture to survive. So we like days like today. We're perfect. So I'm watching the weather all the time. It's never quite what I think it's going to be. So we try to put them out and we water them in. So we're very careful on making sure, or put it out right after it rains. It's nice and moist. So we do a lot of that to keep these guys working. Okay, this is what we do with all the nematode things. Your nematode samples, your biologicals, treat them like it's a gallon of milk. They can't get too hot, they can't get too cold, and they're going to spoil. So you have a month to use them, which is longer than your milk. But if you order those nematodes, they're only going to be alive for a month. So you've got to use them when you get them. And when you get them, you're spending shipping because it's going to come refrigerated. Does not need to sit in the mailbox. It's too hot. Got to put them in the refrigerator. So as soon as you get them, put them in the refrigerator. And once you decide to use it and you dissolve it in the water, put them all out because they're not going to live after that. You've got to put them out. They won't sit in the solution in your sprayer. So use them all, put them all out. That's how they work. Anything else? Yeah, don't leave them in your truck. So just like if you're going to the grocery store and you're going to be in a few places, you take your ice chest to put your milk in before you get home. That's what you do with the nematodes. So treat them like milk. Be careful with them. Then they'll work, because they'll die. Okay, they don't like sunlight or drying out. So Gary and I put a lot of tests out at dusk. So after dinner, we're always going out. The last part of the day, moisture's coming in, you put out your organic biologicals, and they have all night in the moisture to find their nematode or their insect prey and attack, and they'll survive. So it really works well to do it in the evenings. We have some first thing in the morning, and today would have been fine if the weather had predicted and it did what it said it was going to. So if it's rainy and moist all day, that's perfect. But we've had a lot of days they predicted rain and nothing happened. So we tend to go to the evenings.
Or if it's even cloudy all day.
Cloudy all day is great if it does it. Yes, that's perfect too, that works. The nematodes are best 48 to 93. So we can't have any freezes, that's too soon. And if we hit over 93, the soil doesn't usually get that hot, but you're gonna have to water them in. But that's when they're normally their best temperatures. Evening. And then we're always putting it on crops rather than the bees, so this may not matter. We don't fertilize for 2 weeks. So if I'm putting these on soybeans or any other crops or turf, you don't fertilize because the nitrogen will take out the nematodes. But with bees, I don't think that'll matter. You don't fertilize those guys. So it's not a problem for you guys. And, did the video work? Can you see them moving? They're alive. If you treat them like milk. So you can see them moving. So that's that predator-prey. They're going after the insects to take them out. And that's Wendy's videos she made. And so you can see them moving in that dish. And that was one of the cadavers that they opened up to let them release them. So you can see how they work. Good? I can stop it because it does make noise. Okay. So they work. They're not easy. Yes?
Just one quick question. When you put them into the ground, do they have a preference on the host? So if you've got other insects around other than the ones that we're trying to attack, would they have a preference, or do they just go for any?
They go for any. We only test them on our small hive beetles, but I know that we get these out of any of the grubs and the loopers, so there may be some specificity, but I bet they'll go through whatever one they find.
I was just thinking, if you put me in a room with a bunch of donuts, I always go to the maple ones and
Yes.
leave the other ones. So I just wondered.
I expect they do too. But I'm sure they're also after, if there's only a chocolate, you're going to eat it. Yeah. But I do think they're probably specific.
How does this affect your root crops?
My root crops?
Yeah, like sweet potatoes.
Oh, okay. It reduces the number of wireworms that we're working with. So it reduces the damage to the potato. They don't affect plants. So these nematodes only eat insects. Now my root knot nematode, that one eats the plants. So a specific different ones. But the entomopathogenic ones only eat insects, and we put some out on sweet potatoes right now trying to take out the wireworms to make the quality of the crop better.
So do they go dormant then in the wintertime and come back to life?
Yes. So do they go dormant? I was supposed to repeat questions and I didn't do that. Yes, they go dormant. We call it a dipause stage, but yes, or they stay in the egg. So it takes a temperature and a chemical cue to make them hatch, so that they may stay in that old cadaver, or they'll just stay in the soil as eggs, and then the next season they'll come back with the chemical cues of an insect coming by or the temperature warming up and enough moisture. But they won't come all at once, so they're survival like weeds. Some will hatch and some will wait. Oh, thank you.
We thank you very much.
Oh, thank you for having us.